So we now turn to the President's Prize Medal Lecture. And this will be introduced in two parts. One, visually, so I can have a short rest, and then a more formal introduction. And if we could run the video, please. And put the lights down.
We hope that we can provide an inspiration to everybody, not only Russian, but to everybody that has a dream, that has a big dream, and that is willing to work hard to make that dream come true.
Station, this is President Donald Trump. Do you hear me?
We can hear you.
That's great. What would you like to tell everyone listening and watching today, especially young women who are interested in space?
For us, it's just coming out here and doing our job today. We were the crew that was tasked with this assignment. At the same time, we recognize that it is a historic achievement, and we do, of course, want to give credit to all of those that came before us. There has been a long line of female scientists, explorers, engineers, and astronauts, and we have followed in their footsteps to get us where we are today.
Thank you both very much. Have a good time.
Executed the spacewalk plan and mission the same way that I've executed in the past with my male colleagues, and I'll continue to do that if I have the privilege of going out the hatch one more time. So I think that what I will say is that I hope it's changed because now the whole field has been opened up. So we're in sort of a new chapter now where we've crossed that line, and now 2 women have done it. And so hopefully it'll become commonplace, and it won't even necessarily be something that's a big deal down the road. The idea of having the honor of being the first woman to walk on the moon is almost too great to fathom. Of course, it would be a dream of mine and has been my entire life. But for now, I'll just settle for knowing that I'll probably at least know the first woman to walk on the moon. Hint, hint.
Vote 2, starting turn.
So it really meant a lot to me personally to have this first experience for my spacewalk with Christina, but I think that we do recognize it is a historical achievement. It does carry a lot of weight to other people, and for us, when we see that many times in our lives, it does make a difference if somebody can identify with somebody, something about them, some kind of shared experience with them, whether it's something similar to their background or experiences or what they look like or the fact that they have long curly brown hair, who knows. If sometimes that makes it easier to have an inspiration, have a connection and motivation for something great.
And you see that first umbilical separating. And now keep an eye on the one a little bit lower down. Once that separates, we're 15 seconds away from liftoff.
Пуск, бортовое питание.
Auto sequence initiated.
Auto sequence start. Second umbilical separating. T-minus 15 seconds. You can see the engine starting to fire.
Second umbilical.
The first stage ramping up.
Engine turbo pumps at flight speed. Engines at maximum thrust.
And liftoff.
Liftoff.
Oleg Skripochka, Jessica Meir, Hazza Ali Al Mansuri leaping forth from Gagarin start on their way to the International Space Station.
60 seconds flight.
15 секунд.
15 seconds in flight.
Двигатели первой и второй ступени работают устойчиво.
First and second stage thrusters are operating just fine. Copy. Everything is fine on board the Soyuz. We're feeling great. Copy.
We're going to continue to get these translations throughout the flight. Everything's still sounding good with that first stage. You can see the 4 strap-on boosters lighting up the night sky. Soyuz delivering 930,000 pounds of thrust with that first stage, the 4 boosters and the single core engine. You can see them punching through the cloud layer there.
What a great introduction. I don't need to say too much more when you see that, obviously. But it's a great pleasure to formally introduce the 2021 President's Prize Medal lecturer, Dr. Jessica Meir. Dr. Meir received her undergraduate bachelor's degree in biology from Brown University, and she then went on to a Master's of Science degree in Strasbourg in Space Studies at the International Space University. Returning to the United States to work for Lockheed Martin's Human Research Facility at the Johnson Space Center, she supported human physiology research that was undertaken on the Space Shuttle program and also the International Space Station itself. During this time, she took part in the reduced gravity aircraft flights. These are the parabola flights that the astronauts fly to get momentary weightlessness. And then she served as an aquanaut first before becoming an astronaut. And as an aquanaut, she took part in the Aquarius underwater habitat for the 4th NASA Extreme Environments mission. She then decided to go back to university to complete her postgraduate training, where she went to the Scripps Institute for Oceanography attached to UCSD. And at that time for her PhD, she specialized in diving physiology of emperor penguins in the Antarctic. She also studied the diving physiology of elephant seals in Northern California. And not to be outdone by spending too much time in a high-pressure environment, she decided to switch to a low-pressure environment and then go to Canada, to UBC in Vancouver, to work on the Canadian Barheaded Goose, which flies over Mount Everest. Clearly, during that time, she got really interested in the effect of low oxygen systems on physiological control when she was with Bill Milsom at UBC. She accepted a position as an assistant professor at Harvard Medical School at Mass General Hospital, where she continued her physiological research. And in talking to Jessica, the tension between staying in academia and then fulfilling the opportunity to be selected in the astronaut training program was obviously a real dilemma, and when she'll talk about this herself, but in 2013 she was selected as one of the 8 crew members in the 21st NASA astronaut class, and she went on to undergo intensive training as an astronaut. And if we just wind this forward now, she took part in the famous Expedition 61 and 62 launched by the Soyuz rocket, which you have just seen. She is in the Guinness Book of Records with her co-astronaut for the first all-woman spacewalk. She has accumulated over 205 days in space on the ISS, 3,280 orbits of Earth, and she's traveled 86.9 million miles. She has seen a lot. We're particularly delighted that she has taken time out of her busy schedule in her so-called down phase at the moment before her next exciting phase of her career. She has spent time in Oxford because she was awarded the Sherrington Prize Lecture for the Public Understanding of Science. And to add to another accolade for her in the UK, I'm delighted to now welcome and introduce the President's medal-winning lecturer for 2021, Dr. Jessica Meir.
Thank you very much, David. That was a wonderful introduction, so maybe I don't need to speak for as long tonight since I think we've covered a lot of it already. But it really is an absolute honor to be here, first of all at Oxford and now here in London at the Physiological Society. I mentioned this at Oxford as well, but walking down these halls and these streets named after all of these scientists and giants of physiology and other disciplines that I have been referencing throughout my entire career, it is incredibly humbling and such an honor to find myself here speaking to a room at a society such as this one today. So thank you very much for this honor. And what I will do is start off with a brief video as well, following in David's footsteps there of giving myself a break before I even get started. But I will narrate a little bit of that so we can play the volume of the video, maybe balanced with the mic so that I can narrate as we go. And then I'll switch over to a PowerPoint to show you a little bit more that's specific to my journey in transitioning from a physiologist into an astronaut and how that really came full circle for me most recently on the International Space Station. So we can go ahead and roll the video. So this starts out at our launch at the Baikonur Cosmodrome in Kazakhstan, and there you'll see my commander, Oleg Skripochka, a flown cosmonaut, and then also Hazza Al-Mansouri. He was the very first astronaut from the United Arab Emirates. So a really cool part of our mission to have that historic component and a first for that country. They were, of course, incredibly excited. The different stages of the rocket separating, and this picture was actually taken by Christina of the Soyuz approaching from the International Space Station. So a really cool moment that she captured there. And then this is our view as we approach the space station. And you can probably bring up the volume a little bit on the video.
Astronaut Jessica Meir making her first flight to the station.
You can see I was pretty excited to be there, and I don't think I stopped smiling like that for the whole 7 months.
Feels like home already. It's going to be an amazing 6 months.
The best part about being in space is simply being in space and being weightless and floating and just being able to spin around in a circle at a moment's notice after a meal or during any free time. Most of the food that we eat is dehydrated, then rehydrated, as you can see here, or warmed up in the food warmer. But I was fortunate enough to receive an allocation of some fine cheeses in one of the cargo vehicles, so that made for a nice little charcuterie plate for me. Even drinking water in space is, of course, a lot more enjoyable. Exercise is an incredibly important part of our day, as all of you physiologists are well aware. Even more important in microgravity than here, without the constant loading of gravity to keep our bones and muscles healthy. So we had that weightlifting machine, which you saw there. We have a treadmill and then also a cycle ergometer. So we get some cardiac output, but the biggest advantage really is that loading provided by the weightlifting, which is Merry Christmas. We were up there for holidays, so we had to take some downtime as well in order to celebrate. Christmas usually gets all the attention, so I had to throw some Hanukkah flair as well. But of course, the most important aspect of our time on the space station is conducting scientific experiments. And we are doing that pretty much on a daily basis from fields ranging from combustion science, like you see here, even flames, of course, burn differently in space without gravity. We do protein crystal growth experiments we can grow larger and more pure crystals, which has led to a lot of different pharmaceutical developments in order to create those target proteins. There we were just loading some small satellites into the Japanese airlock to launch those in orbit, and here growing some lettuce even, comparing different wavelengths of light, and hopefully having a sustainable food source for future longer duration missions. This is the life sciences glove box, so one of my favorite areas to work as a biologist. Lots of cell biology experiments, bone cells, muscle cells. We did some rodent research, and I'll be mentioning a little bit more about that in the presentation. There's our -80 freezer, so that's how we keep our samples cold before we bring them back down to Earth. As was mentioned, our mission was quite historic for the number of spacewalks that were accomplished, and that was really just happened to be how the schedule was arranged during our mission. We did 9 spacewalks in 4 months, and usually there'd only be 1 or 2 in that time frame.
You're going to do great today. We'll be waiting for you here in a couple hours when you get home. Have a great EVA.
Drew, thank you so much. Thank you, Drew. Thank you so much.
That's Luca Parmitano, Italian astronaut. He's flying the robotic arm, which you'll see Christina on the end of here, moving her into position for the spacewalk. And this really is one of the absolute highlights of the mission. You know, you're outside of the main volume of the space station, and you're really in your own little mini self-contained spacecraft. Everything that you need to keep you alive is there on your back, and you're really just separated from that void and blackness of space by your thin visor. So it is really a profound experience and just an exceptional one to look down at the planet below. So beautiful, so remarkable, and nothing is separating it. You look down at your boots and nothing in between until this beautiful Earth. So how do we get our supplies that we need on the space station? Most of those come up in visiting cargo vehicles. Because the space station is only at about 400 kilometers above the Earth, we can regularly resupply it. And those vehicles come in pretty close proximity to the space station, and then we fly that Canadarm, the robotic arm, to reach over and grab those and bring them into the space station. We actually fill these full of trash during the mission, and then when they separate, they burn up in the atmosphere, with the exception of the SpaceX Dragon, which can splash down and then deliver any hardware or supplies or scientific samples. Of course, the best thing to do when you have any downtime, or the little that we had, was to look at the exceptional view out through the windows. And the cupola really is one of the best viewing areas for us. We do this for our own enjoyment and we take pictures, but we also do a lot of scientific observations here. You know, we now have this continuous human presence on the space station for over 20 years. So we're flying regularly over some of the same sites so we can document photographs. For example, passing over the Patagonia ice fields and the retreat of those glaciers, the unfortunate retreat of the glaciers in that region. We can capture a lot of things from a unique vantage point on the space station. That's lightning at night, and you can see the city lights as well. We're about to come over Africa where it's fairly dark, and then you'll see that's the Nile River in Cairo, and there's Israel to the right. Those are squid fishing boats off of Thailand, and those, of course, are the northern lights. Both the northern and southern lights are readily apparent from the space station. Really extraordinary to see. Unfortunately, those 205 days went by far too quickly for my liking. I would have stayed at least a year if I could have, if it were up to me, but it was time for our spacecraft to depart, so we had to get in it. And this was also a very strange time. It was April of 2020. When we launched in September of 2019, of course, there was no COVID-19, so it was a bit of a shock for us to realize that we were truly coming back to a completely different planet. It's a pretty dynamic ride. After you undock from the space station, you start entering the atmosphere, and of course you're literally burning up. Luckily, you are protected by the heat shield from the spacecraft, and then you touch down in Kazakhstan. The Russian search and rescue forces are there to greet us, and again, a little bit more difficult to organize than usual due to the fact that we had to figure out how to even get our NASA jet over there to get us home with all of the restrictions for international flights. Okay, so with that, we're going to switch over to the presentation. If we could Thank you. And as I briefly mentioned, I've entitled this one Experimenting in Microgravity, full circle for what I should have said physiologist turned astronaut, especially for this crowd, but I think you'll see what I mean as we go through this presentation. This is a picture from the International Space Station. If you recognize the characteristic shape in the upper right there, that's Cape Cod. And this is the area where I grew up in northern Maine. And from the time that I was about 5 years old, my mother said I started saying I wanted to be an astronaut. And I had this kind of scientific curiosity and also this fascination for biology. That was my favorite subject from the time that I was a child. And I think just trying and wanting to understand more about the plants and the animals. I grew up in a pretty small town in northern Maine there with a lot of forest, and so a lot of nature close by. And I think that had something to do with fostering this scientific curiosity and this desire to explore. My first foray into microgravity research was back in 1999 when I was finishing up my undergraduate at Brown University. Unfortunately, they don't have it now, but they did have this program called the Reduced Gravity Student Flight Opportunities. So as an undergraduate student, you could actually design an experiment to be flown on what David referenced earlier, the Vomit Comet, the airplane that flies in a parabola type shape. You achieve about 30 to 35 seconds of weightlessness. So this was the first time that I had worked in microgravity, and it was an experiment that some of us had put together comparing conventional suturing to using what were fairly new at the time, these tissue adhesives like Dermabond in order to close wounds. We figured in space that would be even more advantageous than suturing. So now I'll sort of describe these different transitionary points throughout my life. And the first one I'll say is projects like this one, also my honors thesis at Brown, where I was also working in a lab that had done some work on the space shuttle, so had all of these biology, physiology, and space connections all rolled into one, which of course really appealed to me. And after serving as a student scientist, as was mentioned, I went to the International Space University and then returned to the US, to Houston, where I was serving as a science coordinator. So at that time of my life, I was coordinating the life science experiments that were being performed on the space shuttle and the International Space Station. Here you can see I'm training Spanish astronaut Pedro Duque, who went on to be the Minister of Science recently in Spain. And he and I were working together. I was training him how to use this ultrasound. And so that was kind of this first transition when I was going back to try to understand more about science. And I had always had this desire to do an advanced degree, but at the time I thought, was it going to be medicine or do a PhD? And I didn't quite know the path that I wanted to take, so I figured a little bit more time spent at work at the Johnson Space Center, somewhere that I knew I was really interested in being, and combining that with some biology and physiology. And I had some extraordinary opportunities working there. David also mentioned the NASA Extreme Environment Mission Operations, and that was an exceptional opportunity where I lived underwater for several days with 2 astronauts and a flight director and some other habitat technicians, and I was just a young scientist at the time. So that was truly an extraordinary opportunity for me in this realm of exploration and seeing the world kind of from the other side of an aquarium. But then I still had that desire to do my own science. It was an exceptional 3 years, coordinating other people's science, seeing those experiments come to fruition on the space shuttle and the space station, and having opportunities like more parabolic flights and this time underwater. But I really wanted to ask more questions of my own. And I came across this field of diving physiology of marine mammals and birds that was being done at Scripps Institution of Oceanography by legends in the field like Jerry Koyman and Paul Panganis. And I was so intrigued by this. I really felt like I had found what I was looking for, this combination of real hardcore physiology. Paul, my advisor, was an anesthesiologist and also a PhD researcher. So we were doing things more in the realm of real hardcore physiology that a lot of people that were working with animals didn't necessarily have the expertise or the funding for. So that was a great opportunity for me at the Scripps Institution of Oceanography. And I did my PhD there looking at the diving physiology of emperor penguins in the Antarctic and then also elephant seals. So as you can see here, we were anesthetizing the animals and then implanting different types of electrodes, heart rate electrodes, so EKG electrodes, or even PO2 electrodes. So we had indwelling catheters, which we were inserting into veins and arteries in order to measure
the PO2 of oxygen during the dive. So we could start understanding what was happening with oxygen levels in these animals as they were diving in the Antarctic. And the emperor penguin, of course, is this iconic species that has been known for a lot of its extraordinary behaviors on the ice, but we were really most interested in studying what it was doing under the ice. These animals can dive for 30 minutes on a single breath. So air-breathing, breath-hold divers, just like us, but with this extraordinary breath-hold capacity. And we were putting these types of recorders on the animals to document these behaviors during the flight, during their dives. So you can see in this graph, this is from one of the heart rate studies. The heart rate in this animal over an 18-minute dive got down to 6 beats per minute for a 5-minute period. So not just a very transient decrease there. And these animals are not at rest. They are actively swimming and foraging and catching fish. So really an interesting response to document if we compare that to what an animal would be doing exercising, a terrestrial animal would be doing exercising. So you can see one of the birds diving with one of our recorders on here. And we were also able to document these pressure profiles as well. I also characterized the oxygen hemoglobin dissociation curve for this species. That hadn't been done before, and that was one I was telling David earlier this week that a lot of these instruments that we use, and I'm sure many of you have used in your physiology careers, I was recreating and building these systems with old Wusthof pumps that I had to rebuild from Frank Powell's lab and others at UC San Diego. And I always remarked to my advisor, all of these things that I'm using for my studies are way older than me, all this ancient physiology equipment. But it was really all of those classic devices which were so instrumental in helping me conduct this work. And I was fortunate enough to dive down there as well. So for any scuba divers in the crowd, highly recommend the opportunity, if you ever come across it, to dive in the Antarctic. I also worked with elephant seals. So now you're talking about an animal that can hold its breath for 2 hours. So again, breath hold divers like us, and a mammal just like us, but holding their breath for 2 hours. And some of the things that we found in these studies, I guess I don't have the references up on these slides, not quite as in tune with the academic audiences now, but we published this kind of work in the Journal of Experimental Biology. This one was published in the American Journal of Physiology. And what we showed was that these animals, really they're adapted threefold in order to conduct these exceptional dives. First of all, they have far more oxygen on board, so they have higher hemoglobin concentrations, higher myoglobin concentrations, higher blood volumes, so they're starting off with more oxygen. They use it very effectively, slowing down their metabolic rates and conserving for critical organs like the brain and for the heart. And what we showed with these oxygen electrodes that were indwelling during the dives is that they have an exceptional hypoxemic tolerance as well. So for example, the arterial partial pressure of oxygen in the elephant seal got down to 10 to 12 millimeters of mercury. So well below the threshold where we as humans would experience shallow water blackout or be unconscious at these pressures. So really, this is how we hypothesize they're able to conduct these incredible dives. Then I switched, as David mentioned, the high pressure wasn't quite enough, and so I wanted to explore this other realm and really try to expand my toolbox a little bit as I went on to do my postdoc, and went up to the University of British Columbia where Bill Milsom's lab was working on animals such as the Barheaded Goose and so many others. And my years there were simply some of the best of my life, those 3 years, and that's an incredible department if you haven't ever worked with those scientists. They're very collegial and still one of the most robust remaining centers of comparative physiology left really on the planet. So I felt quite fortunate to have some time working there with Bill. And what we wanted to do was measure some of the exceptional performance of the Barheaded Goose. So this species migrates twice a year over the tallest mountains on the planet, crossing the Himalayas. And we wanted to understand what is it about this animal's unique physiology that allows them to conduct that iconic migration. Now, I preferred, as you can see from my previous work, I really did prefer working on animals in their natural habitats, but sometimes in order to get at some of these more invasive questions, we thought, for example, in this instance, it would behoove us to fly an animal like this in a wind tunnel so that we could instrument it with several different physiological recorders and also measure metabolic rate during these flights. So what I didn't realize at the time was the scale of the project that I was biting off here. And fortunately, we had some other collaborators working on Barheaded Goose. One of my collaborators, Lucy Hawks, is here tonight, and she was working with Pat Butler and Charles Bishop. And so we had a lot of know-how in the field. What we decided to do was to imprint these birds in order to have them be comfortable so that we would be measuring a real physiological response and not just a fear response with this animal. So I became Mother Goose, and I had a flock of 12 goslings that were imprinted on me. They actually hatched in my hand, and that was quite an opportunity, of course, for a woman in her 30s to have this kind of bond with an animal. Unfortunately, at the time that the wind tunnel, that we were ready, these birds grow very quickly. They had their full flight feathers within a few months, and we were ready to fly them in the wind tunnel. Unfortunately, the wind tunnel was broken at that time. So we wanted to make sure that they developed their flight muscle accordingly. So I would take them out near the animal care facility in Vancouver for these rides on the scooter. It was a lot of fun for me. These animals, my babies, were flying next to me, and sometimes so close that their wingtip would be brushing my shoulder. And of course, no science was conducted at this time, but it was still a very valuable part of the project. But we were successful in the end in getting these birds to fly in the wind tunnel at UBC. You can see me off to the left. I'm kind of encouraging the bird to keep flying as its parent. And my undergraduate student is, of course, doing all of the hard work, as I'm sure many of you have seen in your labs as well. As everybody mentioned with the honorary awards, it really is the most important part of it all, are the extraordinary teams that we work with. So this bird is flying at 10.5% oxygen. If you can see, it has a little backpack recorder, so similar to those diving animals, we're measuring heart rate, and in some cases also measuring the partial pressure of oxygen in the blood vessels. And it was trained to use this mask that we made out of a dental mold material so that we could reduce the inspired oxygen. So it's not a hypobaric wind tunnel. Unfortunately, those don't really exist at these types of speeds and at this size. So we made do with what we had, and we were flowing nitrogen into the mask so that we could reduce the inspired oxygen. And of course, we have another tube that it has attached to a big pump, and so we are collecting all of that expired air and analyzing both the oxygen and CO2 to get metabolic rate. And what we found was that these animals were successful at flying at 10.5% oxygen, so half of what we have here at sea level, of course, equivalent to about 5,500 meters, and even down to 7% oxygen, which would be 9,000 meters, even higher than the summit of Everest. So we know from other studies such as Lucy's that these animals do regularly fly around 6,000 meters up to 7,000 meters. And I think what we demonstrated here is that they could even go over the summit of Everest, like these early anecdotal reports of many British explorers claimed. And what we revealed here was that the metabolic rate actually decreased in hypoxia. So we think this helps explain how they're able to conduct these flights. Flying, of course, is the most costly form of vertebrate locomotion. So, trying to understand how they can do that when oxygen levels are so low at high altitude. This was published just a couple years ago in eLife. It took a little bit longer for us to get these results out since I also sort of took flight away from that job and into this one. But if you're interested in reading more, that is published in eLife now. So this brings us to really that next transition, and that is from my time having gone from coordinating other people's science at NASA, moving into doing my own experiments and research on animals, and then eventually becoming the subject myself in perhaps the most extreme of environments. And I kind of look at this as completing the circle where it's now me paying my dues and being poked and prodded in the name of science after all that I did to the animals and to other astronauts before. So that is, as I mentioned in the film, one of the highlights and really the main focus of our work on the International Space Station is conducting scientific research. And what I'll do for this crowd is just mention some of the physiology and the biology highlights that you might be interested in. But as mentioned, of course, we are working in all disciplines from combustion science, protein crystal growth, radiation, material science, you name it, it is all covered on the International Space Station. A few more examples of what I had to sacrifice in the name of science. This is an experiment called Cerebral Autoregulation. It was a Japanese experiment that was looking at cerebral blood flow and any differences in microgravity. So you can see I'm hooked up here. I've got saturation. I have EKG electrodes on, and I have a Doppler probe here looking at blood flow to the brain. And then if we flash back to this picture back in 2003 when I was training Pedro Duque how to use the ultrasound, 17 years later, I was up there ultrasounding myself, and this was a pretty comprehensive study that was a collaboration between French researchers, Canadian, and also American researchers called Vascular Aging. And this was quite interesting as these researchers on previous experiments had shown that there was a thickening and stiffening of the carotid artery walls, so much so that about a 6-month mission was the equivalent of 10 to 20 years of aging. So of course, these are the kind of things that we want to make sure that we understand so that we can keep astronauts healthy and safe and bring them home safely, and will be even more important when we talk about missions like going to Mars that would be something more like a 3-year mission. So trying to understand what is measuring, and again, this is the question, right? The whole animal physiology of trying to document these responses, and then of course, trying to understand the mechanism behind them. So while I was on the space station, I conducted almost 160 scientific experiments as an operator and sometimes as a subject. Drew has a few more than me there, but that's just because he was up there for 2 years longer. So taking a little bit of a look, if anyone is interested here, at some of the statistics, as mentioned, we're doing all types of different science. And I would say, as many of you do, one of the most important aspects there being that international collaboration. The International Space Station says in the name, it is truly international, and there are many investigators from many different countries that are doing research on the space station, and of course, publish or perish. So it is nice to know that these investigations are resulting in publications as well. So now I just wanted to spend a little bit more time focusing on a couple specific experiments that I thought this crowd might be interested in. And really, as many have said before me, looking at those personal connections as well, because it is those interactions that we have together as scientists in society such as this, working in the labs with our students, with our advisors, and that integrated team that really allows us to accomplish what we do. And those personal connections really make the work so much more meaningful. So this was an experiment called engineered heart tissues that I worked on the space station in the life sciences glove box. And so you can see here, this is really looking at assessing cardiac function in microgravity. There had been quite a few experiments that had been done more at the whole organism level, measuring cardiac responses or looking at individual cells, but there hasn't been that much done on the tissue level. So these investigators created these 3D tissue-engineered artificial muscles, and really they were cardiomyocytes derived from stem cells. And so they made these little mini heart tissues that were spontaneously and continuously contracting, just like our hearts would. And you can see their habitat pictured here. So on the space station, although I'm a scientist, we are all doing the same thing up there, whether or not my background is as a scientist or a military pilot. We all get the same kind of training. And as astronauts, we're simply following procedures. These investigators that have dedicated years of their lives to getting their experiment flown don't want me to be changing their experiment. I like to think that as a scientist, I was helping in terms of how we were conducting the experiments or if any problems arose. Here, I'm just doing a simple media change. And this is one of the unique aspects about working in microgravity. The most challenging thing is actually just the logistical problems that we encounter with microgravity. How do you contain everything? How do you ensure that you are getting the same kind of interactions with things like media without gravity? So I'm doing a media change here for these engineered heart tissues, the EHTs, and just using one syringe to inject some new media and sucking out the old media at the same time. And this is a great example of a truly collaborative experiment. And what I wanted to point out specifically was this personal connection that I had. If you flash back to my time as an undergraduate, the very first experiments that I conducted were in a lab run by Herman Vandenberg. And Peter Lee, who you can see pictured here on the right, he was an MD PhD student at the time. So I was working closely in the lab with him previously, again on a similar system, these bioartificial muscles that were being created back then using mostly skeletal muscles. But now, this really came full circle for me, where I was on the space station conducting one of Peter Lee's experiments, and that really made things so much more meaningful for us. And it was almost hard to believe that 22 years earlier, we were working together in a lab at Brown University, and now we were doing that work on the space station. So a little bit more about how they conducted and made these EHTs, if you're interested. And you can see in these bottom videos, they are actually spontaneously and continuously contracting. And what they wanted to do was measure the contractility and any differences that resulted. So these are mounted on these flexible posts, and they have tiny magnets on the ends, and they had electromagnetic sensors underneath so that they could measure the contractility. So here's a picture. So what we were doing those media changes. We were also fixing these EHTs at various time points, looking at mRNA expression, and then also looking at histology, and then bringing them back down to the ground for further study by the PIs. And these, I actually haven't checked recently if they've published this work yet or not, but these were some of their preliminary data slides. But as you can see here, they did find that these EHTs produced significantly less force output in microgravity, and that the microgravity also appeared to induce significantly increased tissue arrhythmia. So again, really important for us to understand these responses in microgravity and the mechanisms behind them to keep astronauts safe. And of course, there are many other parallels and corollaries to disease states on Earth. And this is really true for everything that we do on the space station. It's not just about the health and lives of the astronauts and further exploration. It is also an incredible model because you see these kinds of changes and responses in sort of an accelerated fashion than you would see them on Earth. So lots of different parallels to disease states on Earth as well. The second experiment I wanted to mention was one of the rodent research experiments, and these sometimes get a little bit less attention with animal rights, and I'm sure with many of you that have worked with animals in the past, it can be difficult in the press or to speak about these types of things, but we are doing this work on the space station, and it is very valuable as well. This particular one that I'll mention, Rodent Research 19, was looking at the myostatin and activin pathway. So this very important signaling pathway, of course, involved with muscle degradation in these mice, in humans, in so many organisms. And they wanted to look at this as possible target therapies for astronauts for long duration missions, but also, again, back to applications on Earth, osteoporosis, muscular dystrophy, all of these types of bone and muscle wasting disease. And this experiment was affectionately known as Mighty Mice. So the PI for this experiment, Si-Jin Lee, he actually was the one that discovered the myostatin gene and has been working with this system. He and his wife both have been working with this system for decades. And you can see the dramatic phenotype here. So basically, we had 3 different types of mice on the space station. We had the control mice, then we had myostatin knockouts, and then we had a third group that was being given target therapies before flight and then also by us in space in order to basically just inhibit that pathway. So an alternative to the myostatin knockout. And you can see the dramatic phenotypic difference down to the muscles as well, and also with the bones. So really interesting results. The key here being that these mice were up there, they were not exercising, and they were able to maintain their muscle mass and their bone density and quality because of these changes. So really interesting to think about as an astronaut. You saw in that video how large those exercise devices are that we're using on the space station. When we go to Mars, we will not have that kind of mass and volume available. So we need to start thinking about, one, maybe smaller pieces of exercise equipment that can accomplish all of that. I think that will be quite difficult from an engineering perspective. So we might have to combine that with some of these kind of target therapies in order to keep healthy, safe astronauts. And again, with all of the applications back on Earth. So the whole Mighty Mice thing, I think, got a lot of attention. I was really excited to see that there was a lot of media response to this experiment. And another kind of full circle story, for me as a life scientist, I was so excited to be doing this kind of work in space. Most of our other colleagues actually are engineers. Most of the astronauts have engineering degrees. But Drew and I that were doing this experiment on the space station, of course, my background, and Drew is actually a physician. So we felt like for once our backgrounds were actually being utilized, and of course we performed the best dissections, far better than any of those military pilots or engineers, I'm sure. But it was extremely rewarding for us to be involved in that kind of research. And because of our added interest and our conversations with the principal investigators, we ended up working with them and actually helping them with a manuscript. And if you saw on the previous slide, even being listed as co-authors on this work. So, continuing to publish in any way I can, despite the job change. Lastly, just to highlight briefly a few of the other physiology studies for this crowd, this is a pretty interesting piece of equipment on the space station right now called the Fabrication Facility. And the goal here is essentially a 3D printer to eventually one day print human organs in space. So if you know anything about trying to create an artificial organ on Earth, the problem is often in the gravity environment, you can't quite grow the layers of cells thick enough to properly form those tissues and to eventually form an organ. And in order to do this, researchers have put scaffolding structures in there to try and create that tissue and organ integrity. But those, of course, can be damaging to the cells. So the hypothesis here is that in microgravity, you might be able to grow those layers of cells thick enough to properly form those tissues and eventually an organ. And to think that maybe, still a little bit of a sci-fi type application, but maybe one day we could be 3D printing these human organs in space to be utilized for the shortage of transplant organs here on Earth. So we did some experiments looking at meniscus. There were also some cardiac cells that were being printed in this facility. Another experiment that deserves being highlighted was called fluid shifts. And this was a very comprehensive physiological undertaking. And it has been conducted on the space station for a few years. Drew was actually the final subject for this experiment. So he was a subject and I was an operator. And what this experiment was really trying to look at this upward fluid shift that we have in microgravity. So, of course, in microgravity, we are not having blood pulled down to our feet as we are here on Earth. You have this upward fluid shift and then also a lot of redistribution. So the goal of this study was to better understand those processes, in particular in light of some of the problems that astronauts have been coming home with. And this is I received a lot of attention recently, and there are some changes actually to the anatomy of the eye. Astronauts are coming back with some changes to their vision, and then also a series of different characteristics. It's been called SANS, Spaceflight Associated Neuroocular Syndrome. It's characterized by a vision shift because of a globe flattening, and then also an optic edema, and then choroidal folds. So the choroid, which is supplying the blood supply to the eye, is folded instead of the nice flat layer, and as does the retina. Also thickens, the choroid will thicken, and the retina will also have these folds. So these changes, if you look at them with an OCT, are quite dramatic. And if you saw these in a patient on the ground, they would most likely be associated with some pretty severe symptoms as well. So the interesting thing is that we're seeing this now in astronauts. And one of the interesting things about these types of physiological parameters or medical conditions that are revealed in astronauts is that we're seeing these things because we're looking at a very healthy population. So we don't actually have the comparative data on Earth. We don't measure these types of things. We don't give these types of tests to healthy population here on Earth. So, we don't know how prevalent they are in a normal healthy population, but certainly in certain disease states, they do cause quite significant problems for humans. I actually had the worst case ever documented of SANS, and I had no actual symptoms other than these responses that were measured with OCT and fundoscopy and also tonometry. So we're looking at all of these different systems to better understand the problem. The only lasting symptom that I have is that when I went to space, I actually needed glasses and contacts, and now I have perfect vision. Because my eyeball, if you look at the MRI before flight, is a nice round eyeball, and now because of this globe flattening, it's flattened in the back, which is of course changing the refraction, and luckily for me, this seems to be the only thing that is persisting. So this has received a lot of attention though, because we do want to make sure that it is not going to be causing any long-term deficits or is maladaptive in astronauts. So what we were doing here was taking a suite of physiological measurements and also highlighting this international collaboration. This is in the Russian module, one of the Russian modules on the space station. And this is a lower body negative pressure device, so it's called the Chibis suit, and it's a Russian piece of hardware. So we are able to put the subjects in this and apply that vacuum pressure to bring that blood back down and take those simultaneous measurements, first in microgravity, then with the LBNP device, and then of course before and after the spaceflight as well. So we were getting set up with some equipment here. We were doing various ultrasound measurements. You can see Drew's medical data above the screen there. It's okay, he knows it's there. There's the EKG pattern, and getting some ultrasound measurements in various vessels, doing the OCT here, so looking at the back of the eye and the retinal layer, trying to understand what's happening in the microgravity versus with the LBNP, the vacuum applied, and doing tonometry measurements to look at the pressure of the eyeball as well. So this was, again, really interesting for Drew and I to be doing together. He's a physician that's used ultrasounds a lot in his time in the Army, and as both life scientists, we were really excited to be involved in this study. So lastly, I'm just going to wrap up by telling you about another payload that was flown on board the International Space Station when I was there. The reason why I like to highlight this, it's called the ISS Experience, and it's this camera that you can see here that's installed in this video in the cupola. And this is a camera that was developed by a company called Felix and Paul in Montreal. And it has a series of cameras around it and then one on the top side. And so it's able to film and capture the whole experience on the space station in this 3D, 360-degree virtual reality. And when I first saw some of the things that they had made in the past, they did one on astronaut training as well, and I appeared in that. And I thought, this is going to be pretty cool. I can see why it'll be cool, but I'm not really a video gamer. I'm sure it's, not going to be that impressive. And when I put it on, I was really in awe of the power of this kind of technology because it really does transport you there and is completely immersive. So the reason why I like to end with this is that it has given me this very powerful way of sharing the unique experience that I've had and been so fortunate to have with so many more people in a totally different way. It truly is immersive, and when I put it on now, I am back on the space station. I can look around and see where I ate, where I slept, where I worked, down to every little detail. So if you have the opportunity, if anyone has an Oculus, you can download this content relatively easily, and it is something, there have been 2 episodes released, there'll be 2 more to come, and it is documenting everything from daily life in the space station, the experiments that we conduct, doing exercise, going for spacewalks, even having a camera outside during one of the spacewalks. So highly encourage you to try and see this content. Felix and Paul will also be bringing this to different planetariums and museums too, to have more interactive experiences for people that don't have an Oculus headset. So just wanted to point out a few of these assets. If you aren't aware, a lot of this information is documented on the NASA websites. There's an app called the Space Station Research Explorer, which you can search for by discipline, by experiment, by mission, and if you want to understand and learn more about any of these experiments. And of course, we have very active social media platforms as well. So with that, I'll just show a few parting shots of where we are now. If you recognize this landform, probably most of you do better than I did, but this was a picture that I took from the International Space Station, and really just illustrating these incredible views that we have, and particularly along the coastlines and the water currents and coastal currents. And you can see London kind of underneath those clouds, but that's probably a view you're used to seeing as well. And I think here you should see where Oxford should lie, and just a few other views back towards many of your home. This is at night, of course, and so you can see Great Britain and you can see the coast of France as well. And this is a picture I took of London. So this was one of the first, I started taking this series of cities of night photos, and I felt like I just got really lucky with this one, and then I became obsessed with trying to replicate it all over the world. But it's harder than you think from the space station because you're moving so quickly, you don't even really know what you're looking at, and you're just trying to shoot away when you know you should be passing over this city. The hardest part is then looking at the pictures and trying to figure out which city you've actually caught. So this one should look quite familiar to you. And that is all. That's all that I wanted to talk with you guys about. I don't think that we're going to have questions now, but of course I'll be here for dinner with most of you, and I'm happy to answer any questions at that time.